Hello, everyone, and welcome back to the MCAT Prep Pod Biology Review. I'm your host, Mark, and today we're continuing from where we left off last time, talking about menstruation, eggs, and sperm by diving into embryogenesis and development. This episode covers the incredible journey from a single fertilized egg to a developing fetus, touching on the key stages and mechanisms involved. Understanding this process is fundamental to biology, and it connects concepts from cell division, genetics, physiology, which are all crucial for the MCAT. So, in this episode, we're going to cover fertilization, cleavage, blastulation, gastrulation, neurulation, and we're going to talk about the formation of the germ layers and look at some of the key mechanisms that are driving development. So we're going to talk about specialization, stem cells, induction, apoptosis, and gene regulation. We'll also talk about later fetal development like circulation, gestation milestones, a lot of stuff to cover today. So, if you haven't got a copy of the show notes, head over to reviewmcat.com, get a copy of those show notes. You can follow along with me, or you can review on paper when you get a chance to sit down at your computer. You can also get the flashcards, which are posted for this, that have all the key terms and things that you need to know for the MCAT on it. So, let's get into the episode here. We've got a lot to cover. We're going to start with fertilization, and this usually occurs in the widest part of the fallopian tube. If you recall from the last episode, we talked about the structure of sperm, and the head contains that acrosomal region with the acrosomal enzymes. So these enzymes allow the sperm to penetrate the two outer layers of the egg, which are called the corona radiata and the zona pellucida. Once the sperm penetrates these outer membrane layers, its pronucleus will enter the oocyte, and this fusion triggers the completion of meiosis II in the oocyte. If you remember from the last episode, oocytes are kind of frozen in meiosis just before meiosis II. Now, following this fusion, there's a reaction that's called the cortical reaction that occurs, and that's where calcium ions are released from the smooth ER or the cytoplasm, depolarizing the egg membrane, which increases the metabolic rate, and this causes changes like hardening of the zona pellucida, and that prevents other sperm from fertilizing the same egg. This block to polyspermy is crucial for development, and the depolarized and impenetrable membrane is sometimes called the fertilization membrane. The metabolic rate of this newly formed zygote increases significantly. If you remember, the egg was just kind of chilling, it wasn't doing anything. Now it's going to start doing metabolism and growing. It's worth noting here that typically during ovulation only one egg is released, but there are instances where two or even more eggs are released and they can both get fertilized. In the case where two get released and they both get fertilized, you get what's called dizygotic twins, and these are the non-identical kinds of twins. In the case of identical twins or monozygotic twins, that's where one egg is fertilized and that splits into two zygotes early in development and both zygotes develop. These kinds of splits can be partial or incomplete, and if that's the case, you end up with what are called co-joined twins. So moving on, after fertilization in the fallopian tube, the zygote will move into the uterus, and it's going to be undergoing rapid mitotic cell divisions along the way, and this division process is called cleavage. A key feature is that cells divide rapidly without significant overall growth between divisions, so the resulting cells, which are called blastomeres, become progressively smaller. So the cells aren't growing in size, they're just dividing. And this increases the surface area to volume ratio, which is going to allow for better nutrient and gas exchange with their environment. If you remember, smaller cells have larger surface area per volume, so they can exchange gas with their environment a little bit easier than larger cells. So cleavage can be either determinate or indeterminate at this point. An indeterminate cleavage is where the early blastomeres retain the potential to develop into a complete organism if separated, and a determinate cleavage is where the fate of the blastomere is set, meaning the cell is already committed to becoming a specific cell type, or maybe even a certain organ type. So, indeterminate, its fate is not determined, it can become anything, even a new complete organism, and determinate, its fate is determined, and it's going to become something specific. So through cleavage, the embryo is going to develop into a solid ball of cells, which are called a morula, and this is about the size of a mulberry. Not that most people know what a mulberry even is, but for some reason it's called a morula, and that's another word for a mulberry. And Anyways, around days 5 to 7, the morula undergoes blastulation. And this is transforming into a structure called a blastula or a blastocyst in mammals. And the blastocyst consists of three parts. It consists of a hollow, fluid-filled cavity called the blastocoel, an outer layer of cells called the trophoblast, which contribute to the chorion and later the placenta, and a cluster of cells inside called the inner cell mass, or the ICM, which are going to give rise to the embryo itself. So during blastulation, you start to develop these specific different things. One's going to be the cavity, one's going to be the placenta, and the other one's going to form into the embryo. Now, around this time, the blastocyst reaches the uterus and it begins implantation into the endometrium. If you remember, that's the uterine lining that we talked about last time that estrogen establishes and progesterone protects. That's the endometrium. The trophoblast cells, which remember are the outer layer, facilitate this implantation and they start forming the chorion, which is a membrane that develops these chorionic villi, and these villi stick out from the chorion and they penetrate into the endometrium, which makes a connection that's eventually going to become the placenta for nutrient, gas, and waste exchange. And it's actually possible at this stage for the blastocyst to implant somewhere other than the uterus, such as the fallopian tube, and this ends up in something called an ectopic pregnancy or an angular pregnancy, which is a dangerous condition for the mother, and it usually ends up in a miscarriage or it requires special medical intervention. So after implantation, other important extraembryonic membranes develop. So there's the yolk sac, which supports the embryo initially, and this is the site of early blood cell formation. The allantois, which is involved in early fluid exchange, and this becomes part of the umbilical cord, and the amnion, which surrounds the embryo, creating the amniotic cavity that's filled with amniotic fluid, which is necessary for shock absorption. And the yolk sac and the allantois remnants are going to contribute to the umbilical cord. And this is what connects the fetus to the placenta. And the umbilical cord typically has two arteries that carry deoxygenated waste and blood away from the fetus, and one vein that carries oxygenated blood and nutrients towards the fetus. So keep in mind that the oxygenation status of these arteries and veins are not typical. So we'll get to the respiratory system soon, but usually veins have deoxygenated blood and arteries have oxygenated blood, but the ones in the umbilical cord are important exceptions to that rule that you have to be aware of. So in the umbilical cord, the arteries have deoxygenated blood and the vein has the oxygenated blood. So following blastulation, the next major step is gastrulation, and this is typically in week 3, and it involves a dramatic reorganization and migration of cells within the blastocyst to form a 3-layered structure called the gastrula. So gastrulation creates the gastrula. Now during this phase, there's a primitive gut tube that gets formed by invagination in the gastrula. If you imagine kind of pushing your finger into a ball of dough to form a pocket in the dough, a similar process is occurring in the gastrula, where a region of cells fold inward and the opening that's formed is called the blastopore. This primitive gut is called the archenteron, and in deuterosomes, like humans, the blastopore develops into the anus. And this process establishes the 3 primary germ layers, which are the foundational tissues from which all the body parts come from. So these 3 layers are first the ectoderm, which is the outer layer, and this will eventually become the epidermis, so your skin, hair, nails, sweat glands, nervous system, brain, spinal cord, peripheral nerves, lenses of the eye, the inner ear, adrenal medulla, pituitary gland. So that's the ectoderm, and you can think attractoderm, and those are things that attract us to others. So skin, eyes, your brains, things that make someone attractive, ectoderm. So the second germ layer is called the mesoderm, and that's the middle layer, and this develops into the musculoskeletal system, so your bones, muscles, cartilage, your circulatory system, heart, blood vessels, blood cells, excretory system, which is your kidneys and ureters, gonads, connective tissues, adrenal cortex, and you can think mesoderm, which is the means of getting around, like your muscles and your bones, your means of getting around in the body, like your circulatory system, and your means of getting around, if you know what I mean. Think gonads. Now the last layer is called the endoderm, and that's the inner layer. This forms the epithelial lining of the digestive tract, the respiratory tract, and the lungs, as well as the associated glands like the pancreas, liver, thyroid, parathyroid, and parts of the bladder and urethra. So you can think the inner organs, the inner derm, endoderm is your inner derm, and that's the linings of the internal passageways, inner organs. So you can think inner derm for this, endoderm, inner derm, that's your organs, linings of internal passageways, inner organs, inner derm, endoderm, whatever. There's one special note here to be aware of for the MCAT, and that is the adrenal gland. It has a dual origin. So the adrenal cortex comes from the mesoderm, and the adrenal medulla comes from the ectoderm. So two parts of that adrenal cortex come from two separate layers. So let's move on to neurulation, and that's the forming of the nervous system. So soon after gastrulation, we start to develop a rudimentary nervous system, and this is going to develop from the ectoderm, like we mentioned before, and the process is called neurulation. So during this process, first the notochord, which is a rod of mesodermal cells along the dorsal axis, is going to induce the overlying ectoderm to thicken into what's called a neural plate. So it starts to develop this thick neural plate. The neural plate is then going to fold inwards, which is going to form what are called the neural folds that are going to surround a neural groove. And these neural folds are going to fuse dorsally, which are going to create a hollow neural tube. And this tube is going to sink below the surface, and that's going to develop into the central nervous system, which are the brain and the spinal cord. And the cells at the tip of the neural folds are called the neural crest cells. These will detach and migrate through the embryo, and these are going to differentiate into a wide variety of structures, including most of the peripheral nervous system, or the PNS. So that's going to be your sensory neurons, your autonomic ganglia, your Schwann cells, melanocytes, calcitonin cells, and parts of the cranial facial skeleton. We're going to talk about the PNS in detail in the next episode, so we're not going to go into that here. I know these developmental stages might be a little confusing just to hear through the audio format, so I would definitely recommend getting a copy of the show notes. I have some figures with this in it, and I'm also going to put a video linked on the website that's going to show this process visually of blastulation, gastrulation, and neurulation, so you can hopefully get a better idea of how everything looks and visualize it a little bit better. So all of these early development stages, especially gastrulation and neurulation, are highly sensitive to disruption by teratogens. And I think I'm saying that word right, but don't hold me to it. And these are agents like alcohol or certain drugs. They could be viruses or bacteria or chemicals that can cause birth defects. Also, dietary influences like insufficient folic acid intake by the mother can increase risk of neural tube defects like spinal bifida. And also maternal conditions like poorly controlled diabetes that can also affect development at these stages. So now that we've talked about these development processes, I'm going to talk about a few key mechanisms of development. And there are a few mechanisms we need to understand when it comes to embryonic development. These are important for us to understand when thinking about how cells know what to do during development. So the first thing we're going to talk about is cell specialization, and this involves two steps. So the first step is called determination, and this is irreversible commitment of a cell to having a particular function in the future. So the cell hasn't changed yet, but its fate is sealed. And this involves specific signaling pathways that become active, or some kind of asymmetrical distribution of cytoplasmic factors during mitosis. And then the second step is differentiation. And that's the process by which the determined cell actually changes its structure, function, and biochemistry to become a specialized cell type. So that'd be like a muscle cell or a neuron, and those are the two steps in cell specialization. So determination, differentiation. So another key mechanism of development is called gene expression and regulation, and this differential gene expression is one of the things that drives determination and differentiation. So since all of these developing cells are going to have the same genome, only specific subsets of genes are going to be active that are undergoing transcription and translation in particular cell types. So this selective expression is controlled by transcription factors, and it's influenced by epigenetic modifications, and these are DNA methylation and histone modifications that regulate gene accessibility without actually changing the DNA itself. Now, I want to take a quick side note and mention histones here. We've mentioned them several times, and in case you aren't familiar or you don't remember what they are, these are proteins inside the nucleus that are involved in DNA storage. DNA is wound around these proteins so it can be effectively stored and regulated. We're not going to go into mechanisms for the MCAT, but it's important to know what they are and that they can be modified to prevent DNA accessibility, which is important for gene regulation. The next important mechanism of development is stem cells, and these are undifferentiated or partially differentiated cells that can differentiate into various cell types and proliferate indefinitely to produce more of the same stem cell. These are crucial for development and tissue maintenance, and their potential for differentiation is described by a term called potency. So there are totipotent stem cells, and these can differentiate into all cell types, including embryonic and extraembryonic or placental tissues. And these are only found in the earliest stages, so the zygote, the early morula. As things develop, these turn into pluripotent stem cells, and pluripotent stem cells, they can differentiate into all cell types of the embryo. So you can think the germ layers, but not extraembryonic tissues. These are found in the inner cell mass of the blastocyst. And then there are multipotent stem cells, and these can differentiate into a limited range of cell types within a specific tissue or lineage. And these are also found in adult tissues. So for example, you have hematopoietic stem cells that can produce any kinds of blood cells, but the cells are limited in what they can actually differentiate into. So just to go over those one more time, there's totipotent stem cells. Those can go into anything, including the placental tissue. There are pluripotent stem cells, which can differentiate into all the types of cells that the embryo needs. And then there's multipotent stem cells, and those are within a specific cell tissue type. So the next mechanism of development we're going to talk about is cell-to-cell communication and induction. And so cells don't develop in isolation. They're constantly communicating with each other. And induction is where one cell or group of cells, which we're going to call the inducer here, is going to influence the development or fate of the neighboring responder cells by releasing some chemical signal called inducers or morphogens. Now, the responder cells have to be competent to receive this signal, and competent meaning be able to understand the signal and be able to differentiate. So this signaling can occur by a few different mechanisms. There's what's called autocrine induction, and that's where the cell actually signals itself. There's paracrine induction, where the signal acts on a nearby cell. And then there's juxtacrine induction, which is a direct cell-to-cell contact signaling. And then finally, there's endocrine induction, where the signals travel through the bloodstream using hormones. And this is much more common later in development, and it's going to be a big focus of this podcast series in later episodes. And remember, we already saw some endocrine signaling in the last episode. And then cells can also communicate using growth factors, which are specific peptides that promote growth, differentiation, and mitosis in certain cell types. Now, the last developmental mechanism we're going to talk about here is apoptosis, or programmed cell death. And this is essential for normal development. It's a very controlled process that eliminates unnecessary, damaged, or infected cells without causing inflammation. And it helps to sculpt the tissue shape. So, for example, fetal fingers and toes, they're actually webbed, and this webbing between them gets removed by apoptosis. So cells undergoing apoptosis shrink and form membrane-bound apoptotic blebs, which are then phagocytosed and recycled into other cells. Okay, so this brings us to the last section here in embryonic development, which is the later development. So the fetal stage, circulation, and gestation. So following embryogenesis, which is roughly the first 8 weeks, you get into the fetal stage, and this is characterized primarily by growth and maturation of existing organs. Now, fetal circulation is pretty unique because the lungs and the liver aren't yet fully functional, so gas exchange and waste removal are all going to be handled by the placenta and the mother. So in the placenta, Nutrients and oxygen are going to diffuse from the maternal blood to the fetal blood in the placenta, but the blood doesn't mix here, so it's diffusion across membranes here. This is aided by fetal hemoglobin, or HbF, which has a higher affinity for oxygen than adult hemoglobin, or HbA. So because of this, oxygen is going to transfer from HbA to HbF across this membrane. Now the umbilical cord itself, like we mentioned, has two arteries, and these carry deoxygenated blood and waste away from the fetus to the placenta. It also has the one umbilical vein, which carries oxygenated blood and nutrients from the placenta to the fetus. And if you remember my note from before, the umbilical arteries and vein are opposite of the general rule of thumb for oxygenated status of blood. So one more time, usually veins have deoxygenated blood and arteries have oxygenated blood, but in the umbilical cord, it's the arteries that have deoxygenated blood and the vein that has oxygenated blood. So in the developing fetus, there are some organs that are bypassed by the blood with what are called fetal shunts. And there are 3 fetal shunts that bypass the lungs and the liver, and these are called the foramen ovale, which connects the right atrium to the left atrium, and it bypasses the lungs because, remember, the fetus doesn't need to breathe at this point. The ductus arteriosus, which connects the pulmonary artery to the aorta, and this also bypasses the lungs, and the ductus venosus, which connects the umbilical cord to the inferior vena cava, bypassing the liver so the baby relies on the mother for detoxification. And these shunts normally close after birth. So human gestation or pregnancy lasts about 280 days or 40 weeks, and it's divided into 3 trimesters. So in the first trimester is where the major organogenesis occurs. The heartbeat begins, limbs form, the brain starts to develop, and by the end of this trimester, the embryo is considered a fetus. This is also the most sensitive period for teratogens, which are things like drugs and alcohol that might affect development. And at the end of this trimester, the fetus is around 9 centimeters long. Now in the second trimester, there's rapid growth, movement begins, the face starts to look actually human, the digits elongate, and at the end of this trimester, the fetus is around 30 to 36 centimeters long. Then in the third trimester, there's continued rapid growth and brain development, and then there's a transfer of antibodies from the mother to the fetus to prepare it for birth. There's actually less movement here due to space constraints, and the fetus will reach around 50 centimeters. And at the end of the third trimester is when birth happens, or parturition. And that's when, ready or not, baby's coming, and this involves 3 stages. So first, the cervix thins and dilates, and the amniotic sac ruptures, and this is commonly called the water breaking. This is followed by strong uterine contractions that expel the fetus, and then finally the placenta and umbilical cord are expelled, which are commonly referred to as the afterbirth. And then just when you think it's over and you can take a break and rest, you realize the challenge is only starting. But we're going to get into some of those challenges that come after birth during the psychology section. As for now, we're going to wrap up the episode on embryonic development. I know it seems like the main focus was just on the early stages and we kind of skipped the whole last 2 trimesters, but that's kind of how things are weighted for the test, and I really want to make these episodes super focused on mostly things you need to know and the occasional corny joke, which, let's be real, you also need to know. So, let's do a quick key terms and concepts recap and finish up. This was a pretty packed episode, but here's a quick summary of the key concepts you need to remember for the MCAT. We started with fertilization, which usually happens in the fallopian tube. The sperm penetrates the egg using the acrosomal enzymes, triggering the cortical reaction, which is a calcium-dependent block to polyspermy, and then meiosis II is going to complete in the oocyte. The zygote's going to undergo rapid cleavage, dividing into smaller and smaller blastomeres without growing, forming a morula, and then undergoing blastulation into a blastocyst with this fluid-filled cavity, the tropoblast, and the inner cell mass. The tropoblast is going to contribute to the placenta, while the inner cell mass becomes the embryo. Next is implantation, where the blastocyst is going to embed in the endometrium. Trophoblast cells are going to form the chorionic villi, which are going to help establish the placenta. And we also introduced extra embryonic membranes here, the amnion, the yolk sac, allantois, and the chorion, and these are all vital for support and exchange. We also talked about ectopic pregnancies, which are abnormal implantations and are medically dangerous. Then in gastrulation, we saw the formation of the 3 germ layers, the ectoderm or the atractoderm, which is nervous system, skin, lens, adrenal medulla, the mesoderm or the mesoderm, getting around for your muscles, bones, blood, gonads, adrenal cortex, and the endoderm, which is the inner derm, your gut lining, lungs, liver, pancreas. And remember, the adrenal gland sits at the barrier between the ectoderm and the mesoderm. So the medulla is in the ectoderm and the cortex is in the mesoderm. Next came neurulation, where the notochord induces the ectoderm to form the neural plate, which folds into the neural tube, which later becomes the central nervous system. And it also releases neural crest cells, which form parts of the peripheral nervous system. This is where neural tube defects like spinal bifida and encephaly can occur, especially if there is a folate deficiency. We also discussed key mechanisms for development. First, we had determination and differentiation. We also had gene regulation with transcription factors and epigenetics, including histone modifications. We talked about stem cell potency, so there's totipotent, pluripotent, and multipotent. We talked about the types of induction, which were autocrine, paracrine, juxtacrine, and endocrine. And we talked about apoptosis for tissue sculpting, like separating fingers and toes. Then we transitioned into fetal development. So the fetal stage begins around week 9. It focuses on organ growth, maturation. Fetal circulation relies on the placenta, with the oxygenated blood being delivered by the umbilical vein and waste being removed by the umbilical arteries. Note the reversal in oxygenated status here. And we talked about the 3 fetal shunts that bypass the underdeveloped organs: the foramen ovale, the ductus arteriosus, and the ductus venosus. And then finally, we wrapped up with the trimesters of gestation. So first trimester was organogenesis, heartbeat, neurodevelopment, highest sensitivity to teratogens. The second trimester was growth, movement, facial features. The third trimester was brain development, antibody transfer, limited movement. And then we talked about parturition, which is birth. And the 3 stages we talked about for birth were cervical dilation, fetal expulsion, and then the delivery of the placenta or the afterbirth. And that's going to wrap it all up. I know we talked about a lot of stuff here, and some of it I really recommend looking at the diagrams and the video that I put on the website. Hopefully that's going to be very helpful to you in understanding all of this stuff. But come by the website, reviewmcat.com. I know I introduced a lot of jargon in this episode, and hopefully I said things correctly, but if anything really bothered you, feel free to email me, mark@reviewmcat.com. I'd love to hear from you. I'd love to get some feedback on the episodes, so if you didn't like something, then just email me. If you loved it, then please leave us a 5-star review wherever you found us. That way other people can find us, and it keeps me motivated for putting out more episodes. So, I'm glad you studied with me today. Thank you for dropping by, and I'm hoping that I'll get to catch you in the next episode. Alrighty, take care.